presentation today will be given by two experts, Ibrahim Jivanjee and Rami Zahr. Ibrahim has his bachelor's degree in molecular biology from the University of California at San Diego, and his master's in business administration from the Henry B. Tippie School of Management at the University of Iowa. Prior to working at IDT, Ibrahim has worked in product development in the fields of proteomics, molecular therapeutics, and diagnostics. His role at IDT is in the development of next-generation sequencing solutions. Rami Zahr has his bachelor's degree in chemical engineering from Cornell University and his master's of science in bioinformatics from Johns Hopkins University. He has previously worked at the NIH studying the immune system development in rabbits, mice, and humans, and he is now an NGS Field Applications Specialist for IDT. Ibrahim and Rami's presentation today should last about 30 minutes, and following the presentation, we'll take time to answer questions from the audience. As attendees, you've been muted, but we encourage you to ask questions at any time, and you can do that by typing them into the questions box, which you will find located at the right-hand side of your screen in the GoToWebinar software. And if you click on the little box, there's either a little arrow out or a little plus sign. You can pop that out and then make it bigger to type into it. In case you need to leave early or want to revisit this webinar later, we are recording the presentation and we'll make the link available for the recording a few days after the presentation. That will be found on our website, www.idtdna.com, and you can find that under the support tab in our video library. We have a whole bunch of other webinars there that we've done previously, and also on our YouTube channel at www.youtube.com/IDTDNA bio. So at this point, I'm just going to hand everything over to Ibrahim to get started on his presentation.
Thank you, Hans, and thank you everyone for joining us for this webinar. We are really excited to be here to tell you a little bit about some of the great things that we're doing in the field of next-generation sequencing. My name is Ibrahim. As Hans mentioned, I'm the next-generation sequencing product manager, and I'm going to start off the presentation just providing a brief introduction on next-gen sequencing on IDT and what our role has been in this space. So as you know, sequencing technology has been around for decades. However, the modern next-generation sequencing technology, as you know it today, I think traces itself back to the beginning of the Human Genome Project, in which the funding of the first human genome, which started in 1990, which took 13 years and $3 billion, is what really catalyzed advances in development of at least attempting to develop new and higher throughput sequencing technologies. It was during this time that technologies such as Sanger sequencing and capillary electrophoresis sequencing were introduced and utilized. And it was in 2004 we started seeing the first next generation sequencer, which was the 454 sequencing platform, which did sequencing by pyrosequencing. What we saw in 2005, between 2005 and 2010 or so, we saw the introduction of new sequencing technologies, intense competition, unprecedented level of technological development, which really drove down the cost of DNA sequencing from tens of thousands per megabase of DNA sequence to pennies per megabase of DNA sequence. And so now, after about 2011, we started seeing a plateau in the cost of DNA sequencing, and really the emphasis from a commercialization standpoint from all the companies that have been working in this field is not to necessarily drive down the cost of DNA sequencing as much as making this technology available to researchers in every field of life science through the introduction of benchtop platforms such as the Ion Torrent or the Illumina MiSeq. So that brings us to where we are today, in which this next generation sequencing technology has essentially permeated through every field of biological research, whether you're doing marine biology research, understanding microbiome in the gut, personalized medicine or drug development, and also in screening for crops and agriculture for desirable traits. Next generation sequencing has proved to be a valuable tool, regardless of the type of research you're doing, as long as where understanding the genetic basis of any organism can add value to your research. So IDT has been involved with the since the beginning of next generation sequencing, even before when Sanger sequencing was the method of choice, IDT has been around to provide valuable solutions to help researchers accomplish their goals. With next-gen sequencing specifically, we have been working with thought leaders to understand and address distinct challenges. A lot of some interviews with some of these thought leaders are featured in our Decoded newsletter. Some articles that I think I would like to point out are our interview with Dr. Elaine Martis from the Genome Institute at Washington University, our interview with the Foundation Medicine, which is one of our early adopters of our target capture technology. And there's many interviewers from many researchers from around the world who've worked with IDT to overcome unique challenges associated with next generation sequencing. You can find these articles on our website, searching NGS Your Research in the search box on idtdna.com, or you can check out our Decoded newsletter at idtdna.com/decoded. So what IDT brings to the table in the next-gen sequencing space is our knowledge of nucleic acid synthesis. So IDT is a leader in the development and manufacture of custom biology products, namely DNA and RNA custom manufacturing. We were founded in 1987, and we've grown to be the largest custom oligonucleotide manufacturer in the world. And so our goal for next-gen sequencing is to leverage our knowledge of DNA synthesis to provide the highest quality, least biased scientific results possible. And not only that, but to offer the greatest level of flexibility and customization. So we want to ensure that good science is produced. A lot of these platforms have inherent biases. Some of the workflows involved with generating that data, those workflows have biases. And so we are working very hard to minimize that bias and also reducing the complexity of those workflows so that we can make this accessible to all levels of experienced researchers. And we also try to offer different pricing scales so that we can scale with you to make sure that our products can scale with your needs. So some of the products that you'll hear about in this presentation that Rami will discuss are xGen lockdown probes, which are very high quality, individually synthesized and quality controlled capture probes, which are based on our ultramer synthesis platform. And they are between 60 to 120 nucleotides in length, although we generally recommend 120 nucleotides for any design strategy. And then those products are shipped within 7 to 10 business days. We also have our standard blocking oligo product, which have been pre-designed for easy ordering specific to the adapters that you are working with when you're preparing your samples. And so with this product, you don't necessarily need to buy an entire kit in order to use blocking oligos. You can select only the ones that you need, and this product will ship in 2 to 6 business days. And then we've also provided researchers with a 48-hour capture protocol. This is a do-it-yourself buffers and reagents. We provide all the recipes you need to perform target capture for those who want to have complete control over all the materials in their lab. Over the past 2 weeks, we've actually launched a few new products. The first one that we were very excited about is the launch of a stocked panel, the xGen Acute Myeloid Leukemia Cancer Panel. This panel consists of over 11,000 probes targeting 260 genes that are specific to acute myeloid leukemia. These targets were defined as part of a TCGA WashU Genome Institute collaboration, and those findings were published in the New England Journal of Medicine publication on May 30th this year. These 200 patients who had acute myeloid leukemia underwent whole genome and whole exome sequencing, and these 260 genes were identified as recurrently mutated genes. So this is an extremely relevant panel for AML research and AML type applications. We also launched xGen Universal Blocking Oligos. As I mentioned in the previous slide, our standard blocking oligos allow you to order a blocking oligo specific to the adapter that you are using to prepare your samples. However, many researchers are multiplexing 20, 96, hundreds of samples at a time when they do target capture experiments. And our universal blocking oligos offer a solution which effectively block multiple index adapters with a single oligo without degradation of performance. So you have consistent on-target performance with high multiplex captures, and Rami will talk a little bit about that as well. And then also we've also developed a 4-hour hybridization capture protocol with a 4-hour hybridization time and 2 to 4 hours of hands-on time. This protocol is extremely robust, featuring high uniformity of enrichment. It does depend on nibbles and buffers and reagents, so it's not the type of protocol that if you're a lab that wants to have complete control over all the reagents, this is something that you need to have a kit for that you purchase from a third-party manufacturer. And that is the end of my portion of the presentation. I am going to hand it off to Rami to talk about applications of targeted next-generation sequencing.
All right. Thank you, Ibrahim. And as Ibrahim mentioned, I am the NGS Field Applications Specialist for IDT. And I want to share with you a little bit about some of the applications for targeted enrichment, especially ones that may not occur to people right away. It's just some new applications of ways you can extract more from your experiments using lockdown probes. So I think most people are familiar with the way whole genome sequencing works, just the general product flow. And that begins basically with genomic DNA and proceeds with fragmentation, and then you have the attaching of adapters that are that work with the either flow cell or emulsion PCR or whatever sequencing format you're using, and then you proceed with the sequencing. Target enrichment is actually focusing only on a subset of the genomic DNA, so you're trying to capture only regions that you're interested in learning about. And hybrid capture works off a lot of the workflow that whole genome sequencing uses. It begins with fragmentation. There's attachment of adapters, and then you proceed to hybrid capture, which then you go on to sequence. And there's also amplicon generation for the target enrichment pathway, and that focuses on amplifying specific regions of the genome and then sequencing those regions. So the biggest advantage of using target enrichment is to increase the amount of samples that you can run on using a single sequencing run. So you can imagine that for a large genome, you may be able to do only 1, potentially 10 samples in a single run. But for target enrichment, you're looking at hundreds of thousands of samples in a single run. And this is greatly powerful to researchers that are trying to have enough samples to get statistically significant results, or for diagnostics or such use like that. The other aspect is the target analysis side. So the bioinformatics is a huge part of genome sequencing, and when you have 3 billion bases with a lot of complexities, it's hard to do the bioinformatics analysis. For target enrichment, you can kind of ignore those regions if you're not targeting those regions. So you have a variable target space anywhere between 5 kb to 60 million bases, which is about the size of an exome. And so this gives you some flexibility. It makes the analysis a lot easier. Obviously, because these are different approaches, they have different applications. The whole genome sequencing strategies usually are used for discovery, such as genome-wide association studies, where you're not focusing on a subset of the genome, but you're just unsure of where the mutations will occur throughout the entire genome. Or if you're building a reference, if it's an organism that has never been sequenced before, so you don't have any information about that genome, and so you want to start by building that reference up. For target enrichment, you're looking actually at variant detection and rare variant discovery. So this could be something where you have a heterogeneous sample, and you want to detect if you have a mutation within that sample that might indicate a specific phenotype. For example, if it's a mixture of healthy tissue and cancer tissue, you might be able to detect the mutant that can be treated with a certain type of drug or a treatment mode. And so the target enrichment allows you to get more reads within those regions and detect those rare variants. This is an overview of the protocol, and it basically begins with a prep library, either from an Illumina kit or from another vendor kit, such as like a Ion Torrent PGM kit, for example. And you start with a mixture of targets. So on the very top, you start with a mixture of targets and and off-target regions. So the red here are off-target and the blue are target regions. As you can see, they have already the adapters attached to them, and they're put in solution with blocking oligos, which bind to the adapter sequences. So that those regions don't contribute to any capture of during the enrichment process. So then you add in your capture probes, which are the xGen lockdown probes, and these hybridize to the regions of interest within your sample. So you can see, again, they're hybridizing to the blue regions, which are your target regions, and the red regions do not have any hybridized xGen probes to them. Streptavidin beads are added in so that you can isolate the targets, the xGen lockdown probes hybridize to the targets from the off-target material. The off-target material is discarded, and a few washes removes any spurious interaction between off-target and lockdown probes. And subsequently, you do a PCR step, and then you do a QC and sequencing after that. One thing we wanted to look at is whether you could use these probes to target both alleles. So if you have a mutant that has several mutations, either a single mutation or several mutations, whether you can capture that with the same probe that you're catching the wild type allele. And one way to do this was to look at a hybridization study with a single 120-mer oligo to different amounts of mutations. So we considered 0 to 7 bases of mismatches. So these are the equivalent of substitutions. We looked at a GT pairing, which is a fairly stable permissive pairing. And we also looked at a very disruptive TT pairing, which tends to be less stable and causes a greater differential in the melting temperature. We also didn't want to ignore indels or insertions and deletions. These are, not everyone is studying substitutions. Some people are studying also insertion deletion of bases. So we looked at 1, 3, or 7 base insertions or deletions. So on the top, we have the setup for the insertion deletion experiment. The probe is located on the bottom in blue, and on the top you have the red target. And in this case, we inserted a 1, 3, or 7 base pair insertion that was all T's. On the right-hand side, we did 2 insertions, all T's or C's, and they were on both the 5' and 3' end. And below, we set up a substitution reaction where we have GT or TT on the 5' end, then 1 in the center. And then we tried 3 and 7 as well. So I just want to quickly summarize the data that we got from this. We saw less than a 5-degree TM differential, or melting temperature differential, when we had 1 to 7 base mismatches, when we had 1 to 7 base mismatches in the target. And then when we looked at the insertions, whether it was 1 or 2 up to 7 base insertions, the highest TM differential was less than 4 degrees. These are small changes in TM that, for the most part, will not affect capture because the annealing temperature of these 2 oligos to each other is in the range of 80 degrees, so around 80 degrees Celsius, whereas the hybridization temperature that the probes and targets are incubated at is around 48 degrees. So this huge difference between the 2 will make it unlikely that any of this will affect any of these mutations will affect capture of the mutant alleles. So a single 120-mer capture probe will be sufficient to capture both the wild type as well as the mutant allele. I wanted to go over some of the applications that you can use these probes for. Some of the first 2 I think are less obvious, and I wanted to focus on those because I think they're very exciting applications of xGen lockdown probes. I think they fit well with a lot of the needs with the xGen lockdown probes uniquely fit the needs of customers for these applications. So the first I'd like to go over is the identification of integration sites of transposons or viral genomes. The second is the capture of novel translocations or recombinations. So these can be either full long chromosomal locations, such as like a BCR-ABL fusion, or it could be a VDJ recombination for antibody profiling, or in some cases it can be splice variants, where you're looking at either the inclusion of an extra exon or the dropping of an exon, and being able to capture those events. And lastly, I think we'll cover, the area that most people are aware of, which is identifying SNPs or mutations in regions of interest. So the first application is that of viral and transposon integration sites. A lot of cases when people are studying viral and transposon integration, they're unsure of where these DNA elements are going to integrate into the genome. And a lot of times it's important to determine where they are because it can either affect function or it could have either detrimental effects downstream on the experiment. So in this case, the researcher knows that the viral genome or the transposon sequence, and what they're interested in is identifying these unknown integration sites that flank the known viral transposon sequence. So the method is really to design probes to capture these known viral and transposon sequence, and once you capture these, to sequence the integration sites. So you can imagine when you are preparing the library, you'll share your DNA, and you'll have some target here being either the viral genome or transposon, with a flanking unknown integration sites. In certain cases, you may not have flanking sites. It'll just wholly be made up of your viral genome or transposon. And in some cases, you'll have the integration site on the 5' end. The probes will capture all of these potentially, and what you'll see is that you can then, once you've captured this and you proceed to sequencing, you can do something like a paired end sequencing experiment where you sequence this end here, which identifies your target or the viral genome or transposon, and then the unknown integration site. So you can then locate where that fragment has integrated into the genome. Same idea applies to the 5' end, where you do sequencing from the 5' end and 3' end to identify this unknown integration site. So translocation recombination has got a very similar feel to it. It's basically, again, you have a region of interest and you want to sequence a flanking region, the region adjacent to the target, and you don't know what that adjacent target is. Again, you're going to design your probes against the region of interest, and you're going to try to find the unknown fusion site, and you're going to try to sequence that site. So there's three ways to approach this. There's a way that's just like the integration site approach, or the transposon site approach, where it integrates and you're just sequencing the unknown fusion site. But what you can also do is look for these fusions if you either are trying to detect them because you know that they exist within your sample, or using it for like a diagnostics approach where that specific fusion event is related to a phenotype. So you can design the probe so that it spans the junction, and you can capture that junction anytime it occurs within your samples. And lastly, if you're doing something like VDJ recombination and you want to know whether a large group of fragments may fuse together, you can have probes that are designed for any of these fragments, and by pulling them down with both probes, you'll identify that fusion junction. The last application I wanted to go over was genotyping. This is a well-known application of sequencing targeted enrichment. And basically what you do here is design a probe over the mutation that you want to analyze. So you have your reference sequence and you have a SNP in the middle, and you just center your probe on top of that SNP. Common question that we get is, why not use PCR? And that makes a lot of sense if you're looking at 10, 20, or maybe even 50 SNPs. But when you start looking at a much greater load of SNPs, if you're looking at genome-wide association studies that have been performed by another lab and you want to validate that type of study within your lab, you're going to be looking at 300 to 500 SNPs, and potentially you'd want to do this for many samples. The probability that something will go wrong is increased. And one possibility is that you've designed your primers against your reference sequence, but in one of your samples you have a SNP located in the hybridization site of your primer. And what we know about that is that primers are a lot more sensitive to these differences in the in sequences, and so a single mismatch in this hybridization site can cause as much as a 7-degree melting temperature differential, and that can either reduce your PCR efficiency or completely cause the PCR to fail. In which case, you either have to re-optimize and redesign the primers from the beginning, or you'd have to find a way around it in a different way. Lockdown probes, on the other hand, don't suffer from this problem because, as we mentioned, it's tolerant to several mutations within the target region. So you can have up to 7 SNPs and potentially more than that within that surrounding your target SNP, the one that you actually want to sequence, without causing any problems. In addition to that, if you decided to expand it to even more SNPs, you don't have to optimize PCR for those additional regions. You can just design another probe to target those regions. I want to follow all that up with the performance of our panel because this will give you an idea of how these probes function together. I've touched on how these probes perform individually to capture different mutants, but it's also important to see how they work in aggregate. And this slide is showing the performance of 2 replicates that were done on at different times. And within each replicate, there are 4 barcodes. So those numbers 10, 11, 17, and 23 on the bottom indicate barcodes, and replicate 1, replicate 2 are 2 different experiments done on different days. So this is the AML panel that Ibrahim talked about. It's a 1.2 megabase panel, and you can see from the fold enrichment, you're getting somewhere around 650 to 700 fold enrichment. And that's a comparison of the amount of reads that you're getting in your target versus the rest of the genome. And then if you look at the average coverage depth, you're getting over 150x. In other words, over 150 reads per base in your target region. And that's enough. That's more than enough to detect SNPs or other variations, and it's also more than enough to multiplex further beyond these 4 barcodes. I wanted to break down how the alignment statistics are done. A lot of people will give you one number for on-target reads. So they'll claim that their on-target reads are about 60% or 50%, and that actually contains a lot of information within it. It's not just unique on target. So what we're showing here is in the bottom part of the graph, that dark green, those are unique on target. In other words, those are fragments that have a unique start and stop site that are within your target region. And you can see from this graph that this makes up more than 33%, or more than a third of your reads are going to be unique and on target. The one in the bar in the lighter green is the amount of on target that are within 500 bases of your target region. As we talked about with the capture of integration sites, flanking regions will be included in the sequencing runs, and so this 20% here indicates that 500 base pair region. So if you add those 2 up, you're talking about over 50% on target if you count the 500 bases flanking. And then there's a small percentage, somewhere between 5 to about 8%, that are duplicates. And we consider anything that's a duplicate that has the same start and stop site. So if your read it starts at the same place and ends at the same place as another read, we consider that a duplicate or an artifact of PCR rather than a unique part of the genome that you pulled out. And the rest of that is off target. So we ran this on a MySeq run, and to give you an idea of what the amount of reads that you'll get from this type of run, you're going to get somewhere around 3 million to about 4 million reads that are either unique on target and that also fall within the 500 base pair flanking region. And if you're interested only on the unique on target, you're going to get 2 million plus reads as unique on target. So it's a large percentage of your reads, especially when you think about that you're subsetting the genome, starting from a large a large genome containing all the material, and you're taking only a subset of that. So you get a lot of depth, and you have the ability to multiplex quite a bit. And these also are numbers with 4 samples barcoded in. So in other words, these are 4 samples put in together that you're getting 2 million reads of. And you have the potential to even barcode further if you don't need as high depth coverage. So another aspect of uniformity, another aspect of the AML performance is uniformity, and this is a little bit of a tricky slide, but I'll try to give an example to make it clear to understand. But basically, this is measuring how much of the reads are within a certain amount of the mean. So if you normalize the mean to 1, how much of the reads are X distance away. So if you imagine that we're talking about 500 X depth, 92% are over 90% are within have 250 X or more, and for about 50% have over 500 X. And this one over here, this is 98% have over 100 X. And it's about 98% or 99% for across the board. So the uniformity between the different barcodes is quite high. When you're talking about 98% having over 100x across all your across your barcoded samples, and then the real impressive number is that for over 90%, you have more than 250x coverage, and that is a lot of coverage, and you'll be able to multiplex a lot if you're covering all the regions that you're interested in. We next wanted to compare our results with our competitors. One really common enrichment panel is the SureSlice panel. So we collaborated with Foundation Medicine to look at the differences between the two. And they had a 133 kb region that they were interested in. So we designed a panel for that, and there's a SureSelect panel that was also designed for that same region. And what we found from that data was that we got about 100% of our regions were covered at 150x or more. For the SureSelect panel, only 80% were covered at 150x or more. So there's a lot of regions that were fall within this area right over here on the left-hand side, and this area is low depth of coverage area. And the importance here is that if you have low depth of coverage, you have less of an ability to multiplex your samples, because if you get 10x in a region, you may not be able to go below that if it's in a region that you're interested in targeting. So you'll just have to do more samples individually rather than be able to multiplex more samples. If all you need is 30x, for the lowest region on the IDT panel, you can multiplex at least 5 samples. We investigated where the differences came up, so we wanted to see why our IDT panel was performing better than the SureSelect panel. And one thing that we saw was there's a decrease in performance of the SureSelect panel as the GC content went up. And so I use this black line to highlight the decrease in the GC content, and you can see that around 50 to 60%, it starts getting really low, and even the decline actually starts between 40 and 50%. Now this region is particularly important because the CDS in the human genome has a GC content anywhere between 47 to 61%, and this is where the SureSelect panel is not performing as well. And if you include the 5' UTR, you're looking at 48 to 72% GC content, and the 5' UTRs are important in gene expression analysis, and so you can see that these are going to be hard regions to capture if that's what you're interested in, and so the IDT lockdown probes are a better alternative to that. I wanted to talk a little bit about the standard blocking oligos that Ibrahim mentioned. As I mentioned, they work early on in the protocol, and they're complementary to the adapter sequences, and they have a 3' modification to inhibit extension. And so the way these work is that they bind to the adapter sequences to inhibit hybridization of adapters to one another. And we have these blocking oligos available for the Illumina, Ion Torrent, and Roche platforms. They can also be used on index adapters as well. And I'll just give a little schematic of how this works. So there are two major issues when you're trying to capture xGen lockdown probes. One is that there's a contribution of repeats even if you don't design your probes to target a repeat. The repeat can be within the fragment that you're capturing, pulling down off-target material. The other problem is with the adapter-to-adapter hybridization. So your adapters are single sequence at this point, so they have the ability to hybridize to another adapter sequence, pulling down this pink region here, which is an off-target DNA region. So the solution for this is COT1 DNA for the repeats. And so when you add this in, they hybridize to the repeats in both your target as well as in the off-target, inhibiting this hybridization so you don't pull down the off-target fragment with your correct on-target fragment. And then you have the xGen blocking oligos, which hybridize to the adapter sequence, inhibiting this hybridization interaction over here, which would pull down off-target material. Remember that all these adapters are available for every single fragment. So the chances that this happens is actually quite high because all the adapters are exposed to pull down off-target material. And this leads to the next slide where we're talking about the efficacy of these blocking oligos, because what you see is a dramatic increase of on-target reads when you put these blocking oligos into your experimental run. So these were two papers that were published that used blocking oligos to improve their sequencing runs. The Blumenstiel paper saw an increase from 34% to 58% of on-target reads. So in other words, the amount of on-target went from 34 to 58. In the Hodges paper, they went from 32 to 64. That's approximate, between the two of them, that's approximately 100% increase of on-target reads. And if you think about how expensive these kits are to sequence, it's really important to make sure that you're getting as much as you can out of them. And so the increased reads can either get you more depth in order to detect those very rare variants, or it allows you to multiplex more samples. So you can go from doing maybe 5 samples at once to 10 samples at once. The universal blocking oligos separate themselves from the standard blocking oligos in that they have the ability to block multiple indices. And this is extremely important, not just because it'll save you money, but also it'll reduce the complexity of your enrichment. Imagine doing an enrichment experiment with 10 to 20 different barcodes, and you have to keep track of 10 to 20 different blockers for each individual barcode. Using a universal blocking oligo, you can block all those barcodes with just a single universal blocking oligo. And we also found that for the 4-hour protocol, these perform better than the individual index blocking oligos or blocking oligos with inosine. And the data for that is shown over here, where we see an on-target for this specific run of over 50% with a universal blocking oligo, less than 40%, just under 40% for the standard blocking oligos with inosines. So this column here is representing the blockers with the individual indices. So if you had 10 barcodes, you'd have 10 blockers with the individual indices. And then on the far right, that performed the worst in this experiment were standard blocking oligos with inosines for barcodes. And if you're unfamiliar with the concept of an inosine base, the inosine base is a base that can hybridize to all four bases. They tend to have some bias, but they do bind to all 4 bases. And we see that that's not enough to compensate for to block all the barcodes that are in your experiment. This is an aplex reaction as well. So in summary, xGen lockdown probes are high-quality, individually QC'd oligos. They enable researchers to identify insertion sites, splice junctions, indels, SNPs. We also have come up with xGen AML panels. The version 1.0 provides a large list of genes that you can either use as a panel if all your genes are found within that panel, or you can customize that panel by adding more genes to it. So if you have 5 or 6 more genes that you want to target, you can design probes for those and add it to the panel. And so you can tailor it to your needs. And finally, xGen standard blocking oligos are used with the lockdown probes to increase on-target capture. The universal blocking oligos block multiple indices and improve the capture, as well as it reduces complexity and cost. So that's all I've got for you today. But there's a lot more information at www.idtdna.com/xgen. There's a lot of decoded articles, as Ibrahim mentioned, that we link to. I know there's a lot of great NGS material coming up also in January in the new decoded, so stay on the lookout for that. And if you have any questions that you'd like to email to us, you can at xgen@idtdna.com. This is a group of NGS specialists who work on NGS night and day, so they should be able to answer your question, or at least point you in the right direction. So that's all, and I'll hand it back to Hans right now.
Okay, so we've got a few questions, but before we get to that, I want to just thank Ibrahim and Rami for their excellent presentation. And if you do have any questions and you haven't already asked them, you can type those into the questions box. You'll find that at the right-hand side of your screen in the GoToWebinar software. You just click the little button in the corner of that bar that pops it out, and you can type your question into that. And I guess we'll just jump right into questions here. I'm going to bring Ibrahim back into this and start with question. This person is most interested in the integration sites of viral genomes in plants, and what they want to know is if IDT is able to assist with the experimental design and or sample planning.
Yeah, I think Rami could help with that question.
Okay, Rami.
Sure. So yeah, we can definitely help. We have not worked as much with the plant genomes as much as we've worked with mammalian genomes, but the concept is very similar. There are some complexities with plant genomes, and we would be willing to discuss those. But some of the issues are, depending on the species, the polyploid nature of the genome. Additional problems are the amount of repeats in some of the genomes. I know the corn genome has got about 85% repeats. So there are some unique challenges to working with those genomes. But we've had people use them for a lot of different use lockdown probes for a lot of different applications and had great success. If you'd like to email us at xgen@idtdna.com, we can talk about specifics for sample prep planning as well as capture issues. So yeah, please email us and we can talk about specifics about that.
Ibrahim, did you want to clarify just a little bit about what we do offer as far as assay designs?
Sure. Yeah. So our standard design process involves just tiling over target regions that can be defined either as gene IDs, gene symbols, or as provided as FASTA sequences. So in the context of trying to detect translocations, if you provide us with FASTA sequences or chromosome locations of your targets, we can easily design against that. Generally, our design process is one business day for a standard design. It might take we'll at least get back to you within one business day, and then we can work with you to ensure that the design meets your experimental goals. We also have a design forum online at www.idt.com/xgen. If you click on the lockdown probe product, you will see a request for design, which has a form which standardizes the format which we accept and readily turn designs around.
Okay. There's a couple of design questions here, so let's see. This person wants to know, if I'm interested in flanking sequences, is there anything that they should do to alter the fragment size used for the library preparation? So Rami, do you have any comments on that?
Yeah, that's a really good question. We generally don't recommend to go too large because that will affect the capture. So I understand why you might want to go larger in order to ensure that the flanking regions are actually incorporated into the fragment with your either transposon or viral genome. But if you go too large, and I think the cutoff is probably going to be, I would start at 600 bases, but you may be able to go up a little bit larger, and I'll see what we have in research. If we've tested a lot larger than that, you could potentially go up to 900 bases and test to see how well that works. But one quick way to confirm that your capture is working well is actually to potentially design a qPCR assay to determine prior to sequencing whether you have good enrichment or not. So there are ways to check to make sure prior to sequencing that you were able to capture efficiently for some of these larger fragments, if that's what you need to capture the flanking region. But probably a good starting place would be 600 bases to about 1 kb, and then higher than that, I think there will be a different change in the chemistry of the hybridization process.
All right, so here's another design question. This person wants to know, if I want to capture / resequence 1 megabase region, how many probes do you recommend and what would be the approximate cost? So that's probably Ibrahim.
Yeah, so for 1 megabase region, we generally do end-to-end tiling for all our designs to start off. For 1 megabase region, that's about, I would say, almost 8,000 probes if I did my math correctly. And so we definitely can synthesize that. And in that situation, we generally offer that as a custom quote. And this is generally economical when you get into a very large sample number range. However, if you are only looking to use this assay for, let's say, 2 or 3 samples, it generally is probably not the most economical approach to go to lockdown solution. However, we do have a lot of experience with supplementing. So in that scenario, if you're only working with just a few samples at a time, it might be worth it to follow an array-based synthesis approach from a different provider. But those approaches tend to have gaps, as Rami showed before. And we have a lot of experience with supplementing those gaps with lockdown probes so that you have even performance across all your targets.
Okay. Ibrahim, can you also say a little bit about the scale of the xGen lockdown probes and what how many reactions people will get out of that?
Sure. So as you know, our process for synthesizing these oligos is very different than what you would have from, let's say, an array synthesis provided capture technology. So I'm going to show you the actual website and where you can go to find our lockdown probes. So if you're on our home page, you can scroll down from products, go to next gen sequencing, and click on target capture. I'm assuming everyone can see my screen at this time. You click on lockdown probes, and what we have is 3 scales: the mini, standard, and XL. And so because we individually synthesize every single probe, the amount that you pay is actually on a per probe basis, and we're not trying to limit you on the number of enrichments that you can do. We just provide you a certain amount of material per probe. So the actual scale or amount of probe varies by each scale. So with the mini scale, every probe you add onto your pool actually adds an additional reaction. So if you purchase 200 mini probes, you actually have 200 reactions. If you have 2,000 mini probes, you have 2,000 reactions. And this approach actually ends up being extremely economical when you are sequencing hundreds and hundreds of samples, or even in a small capture size scale. If you're only working with 10 genes or maybe 500 SNPs for genotyping, this is actually an extremely economical approach because it doesn't involve a large investment up front for synthesizing an entire array of oligo.
Okay. I'm going to come back to a question for Rami then. So in the data that you presented, Rami, you showed that there were roughly 30 to 40% of the reads that were off target. I'm not sure exactly which slide that was, but this person wants to know what would be found in those off target reads. What would those sequences be?
That's an excellent question. I think it's for the most part it's going to be well, it's going to be probably a mixture of things. It could be things that are similar in sequence to the target regions. So for your sequence, the great thing about the lockdown probes is they are tolerant of mismatches. But if you are targeting regions that have high identity to one another, you may be pulling those down as well. So you may be pulling down regions that have similar sequence content to your target. Additionally, when probes no longer have anything to bind to, which can occur if you're doing the smaller panels. So remember, you have 3 billion bases in the human genome, and you're only targeting 1.2 million bases. So you're targeting a very small subset from that. So any probe that doesn't find something to hybridize to, because it just runs out of material to latch onto, will start latching onto other sequences that may have more mismatches or that is less fidelity to the actual target. And so I think you start pulling a lot of those sequences down. I'm not sure if you'll see spikes in certain regions. We're looking into some of that within our own research to see what's contributing to those off targets and how we can reduce that further.
Okay. Here's another question for you, Rami. Then this person wants to know if the xGen lockdown probes can be used to capture longer sequences, and what they're asking is, how would they use the target enrichment to get to sequence multiple genes that are longer, like up to 5 kb?
If you're trying just to sequence longer, but the fragment size is small, you just tile against the entire gene, and that's not really an issue. But if you're asking about whether to capture a very large fragment, so a fragment that's 5 kb or longer, that's a different issue because of the thermodynamics of a probe capturing a region that large. We're starting to look into capturing larger fragments, but we don't have any solid data on sizes that large. I know we've done 600 or so bases, and I know we've gone slightly higher than that, but we haven't gone anywhere near 5 kb yet. But that is an application that we're looking into. Generally, it's for people who are doing longer type sequencing runs. So if you're trying to use techniques or a platform such as like PacBio, where you sequence multiple kbs at once, that could be an application for that, and that is something we're currently investigating. But the chemistry would probably need to change, or at least the conditions would need to change for that capture to be efficient.
And I actually have the opposite question here, Rami, which is, what about sequences that are very short? So if you have a fragment that's only 30 base pairs long, can you use the xGen lockdown probes for those?
Yeah, so we have not used it for fragments that small. There may be the issue of having more off-target material because of that, potentially, just because the probes now are only 120 bases only 30 of the 120 bases are actually capturing something. We could look into reducing the length of the probes. The only other issue I could think of is the GC content of that short fragment is going to play a much larger role than a longer fragment. So the melting temperature of a 30 base fragment is going to be much lower than that of a 60 or 120 base fragment. And so during the wash steps and during the hybridization step, the hybridization between the lockdown probe and that small fragment may not be enough to withstand all the washes. So you probably have to consider using different buffers and potentially a different hybridization temperature to compensate for the short length. And again, it's not something we've tested especially for lengths as short as 30, because you're getting to about a primer length at that point. But if you'd like to explore this further with us, please email us at xgen@idtdna.com, and we can discuss this in more detail.
One quick thing to add to that, if your target space or target size is 30 bases long, what we would, Rami, am I correct? We would actually just tile into the flank surrounding those 30 bases, which would improve the specificity and allow you to capture that 30 base region, and you would sequence inwards, right?
Yeah, absolutely. So if it's not a matter of the fragment length, but it's a matter of your target region, that's not an issue, because we can design the probe to capture some of that flanking region. And some people are worried that by capturing by having the probe aligned to the flanking region as well, that they might be capturing more off target. But in a lot of cases, the fragment that you're sequencing, in all cases, the fragment that you're going to capture and pull down is going to be larger than the fragment that you're interested in sequencing. Consider the fact that the average exome length is about 140 to 150 bases long, and your average fragment length when you shear your DNA is about 600 bases long or 500 bases long. So even if you're targeting a full exon, the difference in size is quite dramatic. So you're always pulling down some flanking region, and I think that's going to be okay for this type of experiment, because then you'll still have the ability to sequence it in most cases.
Okay, here's another application-specific item. This person wants to know if you can capture DNase-1 hypersensitive sites, and they said that the DNase-1 hypersensitive site library is short, again, less than 30 base pairs. So it's, I guess, kind of repeating the same thing. So they can't make the library longer than 30 base pairs.
Yeah, so it's a similar to the previous question. So yeah, you'd run into the same issue of GC content, and you'd probably having to change the chemistry of the hybridization in order to capture those shorter targets.
So I think their best bet would just to contact us and find out what we could do for them as far as helping them with a design, see if we're able to help them.
Yeah, absolutely. I mean, we can, even if lockdown probes are not the solution, we can talk with them and try to come up with a solution for them. We've done a lot of experiments with a lot of different techniques, and so we're well versed in how to work with oligos. So yeah, please contact us, and we'll help you with whatever we can.
So here's a good question for you, Ibrahim, which is this person wants to know what NGS platform people normally use when they use the xGen lockdown probes, and they're asking about the different read lengths for the various NGS platforms. Like if the read length is maybe a reason why it can't be used with some platforms.
I don't think read length is the limitation. We have done extensive development work with the Illumina and Ion Torrent platform. We have used the 250 by 250 paradigm chemistry, along with we've done most of our development work on the 150 by 150 chemistry. And then for the Ion Torrent platform, I'm not exactly sure which chemistry we've been using, but it's the off the 318 chip and 316 chip. I know that we've been working off of that. But as far as what read lengths we've been using for the Ion Torrent, I believe it's the most common one that most people use. So, but to answer the question about read lengths, that is independent of the capture step. That's more about the experimental design.
Okay, one more quick question here. So how well do the xGen lockdown probes work with formalin-fixed paraffin embedded samples? It's a question we've gotten a couple times.
Yep. Rami, you've answered that a few times now.
yeah, we get this question a lot, and I think the distinction needs to be made of where the FFPE samples are actually entering into the workflow, and they enter in in the library prep phase. So if you are able to prepare a good library from your FFPE, lockdown probes don't care where the material came from, as long as the the library quality is high when it enters into the target enrichment. And that was a prerequisite not just for target enrichment, it's a prerequisite for just sequencing in general. So if you're able to get a high quality library to go to target enrichment, you should have no problem at all. Now, I should note that if you are not getting enough material after the library prep, so you prepared the library, you have a good library, but you just don't have the 500 nanograms that you need, you can just multiplex more samples. And that's one of the powerful things about target enrichment, that you can add more samples in. So if you have, let's say, 100 nanograms after library prep, or even 50 nanograms after library prep, you can have 10 samples of 50 nanograms to combine them together to have 500 nanograms of input material into the target enrichment step. So you'd have a barcode for each one to identify which one for which sample you're using, and then pull them together and just start with the target enrichment. And so I think that's the way I think most people think about it. I think if you don't have enough material to do a library prep, then you're not going to have enough library material to do a library prep after enrichment, only because you're going to end up with even less material because you're removing a lot of off-target DNA through the enrichment process. So really the bottleneck here is the library prep and not the target enrichment.
Okay, so we've gotten through most of the questions. There's still a couple left, but we're out of time here. So I'm going to wrap this up. We can follow up with people who did not get their questions answered and follow up with them individually. So at this point, I do want to thank everyone for attending today. We very much appreciate that. And I want to thank Rami and Ibrahim again for their time presenting as well as answering questions. And this is one of a series of webinars that we'll be doing on next generation sequencing as well as other topics. We will email you about these future webinars as they are scheduled. And as a reminder, we have recorded this webinar and it will be on our website at www.idtdna.com. You'll find that in our video library, which is under the support tab. And we also have a YouTube channel where we'll have this video as well as some others. And that is www.youtube.com/IDTDNA bio. So with that, I want to say best of success with your research and have a great day and goodbye.